It is, yeah.
Great. Okay, so I'm gonna be giving a talk on my current PhD work. I'm in my second year of my PhD. I study commensalisms or potential commensalisms in um, generalist arachnid predators in agricultural systems, and I'm currently based out of Newcastle University. So today I'm going to go over commensalisms, crab spiders specifically, the flower crab spiders, my current work, and then a community science project I'm currently running. Um, so to start, I'm just going to briefly go over broadly ecological interactions. So they can be split into two different categories. You've got trophic non-trophic interactions which don't involve consumption, for example, the pollination of a plant, and that in that case is a positive interaction, so that would be a mutualism. And then you have trophic interactions which do involve consumption, like a predator-prey interaction. And in that case, that would be an antagonism, as you've got one species benefiting and the other obviously not benefiting. Commensalisms themselves fall in between that, where you have one species that is neutrally or not impacted, and the other benefits in the context that they're being observed. So, and then you can see where they fit just between mutualism and antagonism there, and how it can. Oh, yeah. So beyond just being neutral and positive, there's two different kinds of commensalism that can occur. Um, so you have true neutral commensalisms where the host is not impacted in any way by the commensal, and then some of my pictures haven't loaded, that's fine. And then the commensal which positively benefits. So an example of this is between pearlfish and starfish and sea cucumbers. Pearlfish actually live inside of the sort of um, internal cavity of starfish and sea cucumbers. However, they just reside in there. They don't have any impact on the species, and they will go out and um, uh, forage for food and so on. So they are only using the starfish for protection. Um, so there's no actual impact. Whereas net neutral commensalism is where the positive and negative impacts of the host neutralize one another, which results in an overall neutral impact. And the commensal still benefits. So this would be an example of this is ants that live on Sonita cacti. So the ants provide a deterrence of herbivores eating the cacti in return for getting a nectar reward. However, the cacti themselves have to produce this extra nectar, which obviously adds a cost to the host. But on top of that, pollinators are also deterred, so then they have a loss to reproduction. And those impacts are so large that they actually counteract the benefit of herbivore deterrence. Why commensalism are important is because they impact multiple facets of ecosystem function and change. So, commensalisms are often facilitating interactions, which means that they allow species to exist in habitats outside what would be within their normal range. Which, in regards to climate change and changes in phenology, can be really beneficial to species that are being quite hit by changes in climate. But on the flip side, it can also facilitate invasive species as well. If you've got this species that provides them with an extra benefit when entering a new system, it can allow them to establish within those systems. Now, I'm working with Misumena vatia, which is the flower crab spider. They're generalist predators, and they are actually able to change their color from yellow to white by producing a pigment called omochrome, and they break it back down to return to white. Um, Now, they are quite easy to identify in regards to our native crab spiders, the Thomistae family in the UK. The females uh, significantly larger than the males, between 6 and 9 millimeters, and they have that kind of very characteristic crab spider large front leg um, autonomy. And some of the females do have red abdominal stripes, and the males are significantly smaller and have quite a characteristic brown striped coloration. For their distribution within the UK, they don't like the north. They're quite ubiquitous throughout the south. And they're currently very active in May and June. That's their primary breeding season. Some of the females have been known to overwinter when we've had quite warm winters, but so generally this time of year, they're breeding quite readily. And I've started to see juveniles out as well, and then the juveniles will overwinter until next year. Um, Now, combining both of those, we know that the flower crab spiders utilize flowers to catch their prey, but we don't necessarily know how they are impacting the host and how they choose their host. So the way that they might fall into a commensalistic interaction with the host is if it's true neutral, they're just sitting atop the flowers, waiting for prey, um, and not having any impact on the flower. However, it could be net neutral if the spiders themselves, by sitting atop the flowers, they could be damaging the ovules and the stamen on top of the flowers. And they might also provide nutrients via the waste that they produce, both through defecation and also any, obviously, they're fluid feeders, so the exoskeletons of the prey that are left behind. The more complicated fact of this is that not all interactions are direct, but they still may have a large impact on the host. So you can have an overall positive impact, for example, if the spiders are preferentially eating pest species, or you can have an overall negative impact if they are preferentially consuming pollinators, beneficials, or deterring them. Um, So it makes the overall interaction profile more complicated, even if the direct interactions between the spider and the host are neutral. So, and then on top of that, how are they choosing the host? It could be due to the prey that are attracted. It could be due to the camouflage itself on the plant. It could also be due to the um, not the individual prey species themselves, but the actual nutrients that they provide. So it might be they are choosing plants that attract species that have high protein or high carbohydrate contents, versus specifically choosing plants that attract specific bee or fly species. Now, my current work, I am in Cornwall right now. I'm working on a 1,000 acre farm that's just based outside of St. Ives, doing direct data collection on the spiders. So I do direct observation and spider collection, and we collect the spiders for dietary analysis, where we do high throughput sequencing of their gut contents. Um, I collect the flower visitors and nectar, so for nutrient assays. And then we're also doing surface DNA samples for the indirect interaction. So this is where we use a swab and a TNAS, which is a buffer in some salty, soapy water, essentially, to pick up any DNA that was shed onto the plant itself. Um, and we're also doing pan traps for community composition data. And then in the lab, identification of the spiders, we do the dietary and eDNA metabarcoding and the nutrient analysis. So I'm going to go into that in a little bit more detail. So from the spiders and the surface DNA, we do DNA extractions, and then we do PCR amplification. So we're using two primers within our PCR amplification process. We're using general primers and spider exclusion primers. Obviously, when you're taking the gut contents of spiders, a large amount of the DNA in that will be the spiders itself's DNA. So we use the exclusion just to pick up on any DNA in there that might be less represented, and it allows for greater amplification of that. And then in this case, we're using nanopore sequencing, and we're doing it in-house at the university. Now, for the nutrition extraction, we do lipid, carbohydrate, and protein assays. So we take the nectar and the prey, and we will essentially dissolve the lipids or the carbohydrates protein, and then from those solutions, we then they undergo a reaction with a reagent that will go a different color depending on the concentration of the different nutrients present. And then we use colorimetric analysis to look at that color change and then assess the concentration. So far, this is just from last year. This is from the direct observations with Misumena at the top in this node, and then the grey lines represent the potential commensalisms with the flower species that they were found on. Obviously, bracken is not a flower. I had one female that was transient and moving between plants at the time, and then the green edges represent flower visitation. So all of the orders along the bottom were visiting all of the plant species in the middle. Then the black line represents direct predation. So this is where when I found females, they were, or individuals, they were directly predating on, in this case, Diptera, so that's the flies. And then the red lines represent the second order interactions. So the hogweed and the red campion have facilitated the predation of the Diptera. Obviously, this is not the nicest diagram to read. So one way to that we're working on reducing the complexity of that is using non-bipartite networks or hypergraphs. So this is where instead of having an edge between two nodes, so between two species, you can have a hyper edge, which allows to represent more real world interactions where you've got those multi-species interactions, such as the spider, flower, and prey, and it links multiple species at once, so it just reduces that complexity. And in our case, we can use it to have the host plant, the commensal, and all of the prey linked at once. Now, I'm 2/3 of the way through my second year, so I've got spiders from last year that are ready to go on the sequencer. So I should have those back by the end of this month. And I've got the nutritional networks and the hypergraphs to produce. So far, I just have the direct observations to discuss. But on top of all of that, I'm also running a community science program, which it's called Spies. We're in the second year. We've had lots of contributions this year and last year, and it will run next year, which is essentially asking members of the general public, the science community, to send in photos of crab spiders in their habitats, and if they're willing to, also then send in samples of the spiders themselves to provide a more regional understanding of what host they're using and whether this differs in different habitats. So whether in urban places, parks, gardens, arable land, does this differ? And if so, what is causing that? And if I have the spiders themselves, then we can also look at whether their diet is different too. Yeah, this is just sort of explaining it. We've asked for a 1 by 1 meter square to of the habitat they are in, just to give us an idea of also what the surrounding plants are and whether this attributes to host plant choice too. And yeah, just the future outcomes of that is we can get that regional difference in behavior and interactions. That is me. If you want to take a look at the community science scheme page, you can scan this barcode. And yeah, that is my talk. My supervisors between Newcastle and Cardiff are, well, Jordan Cuff, Darren Evans, and Vivek Nityanada at Newcastle, and then Fred Windsor down in Cardiff. And you can find my contact all on the right side there.
Rosie, thank you very much. Mind-boggling.
Sorry.
Well, no, you shouldn't apologize at all. Um, and I make a similar comment to what I said to Brandon earlier, which is, some of this is well beyond my level of knowledge. But just where we are in terms of DNA and what you can do with that DNA analysis, it's only by seeing talks such as yours this morning. I think to many of us probably really shows just how much more information we can determine. I'm not going to say with lesser effort, but for example, you can analyze the gut contents of a spider to determine what it's eating, but exclude the results of its own DNA. That was something that I was never aware of. So really, really interesting. Um, Clearly an area that I am never going to get into, but nevertheless fascinating to hear what we can learn. So if you don't mind,
Yeah.
just as we did earlier, um, probably 5, 10 minutes of questions. So if anybody does have any questions, then please put them into the chat. And there's first question here from Alice Bennett West. Uh, a question, do they deter pollinators? Are the pollinators aware of their presence and so avoid those plants? Um,
Um, it's a great question. It's something that we want to look into. So obviously I'm doing the flower visitor surveys. So we're seeing which inverts are visiting the flowers when the spiders are on them. So yeah, it's a great question. And it's obviously, so I'm taking just natural light photos of the spiders, and we are comparing the contrast. But obviously, pollinators like bees often see far more into the um, oh, like UV spectra than we do. So it's something that we want to look into further, but setting up a UV camera setup is quite difficult. Um, but as far as we know so far, they're not deterring um, prey. A lot of my samples are currently coming from invasive giant hogweed, and the flies and other pollinators, wasps and everything are just all over them. They're not bothered by the spiders present there.
Excellent. Question from Barry. Are we seeing much spread north from these species? He's wondering, given the warmer weather driven by climate change, if their range is expanding.
That is not something that I'm specifically aware of. I do think they will be moving further north, but their range is not something that I've been looking into
Yeah.
with my research. But I do think they will be, particularly I'm based out in Newcastle usually, but I've had to come down to Cornwall to do my fieldwork. But I don't have any doubts that in the next few years they will likely be moving further north because they're so broad in the habitats that they use. They're not restricted to very specific habitats. So I can't see a reason why they wouldn't if it is warming.
No, certainly one other member on here would love that to have happened earlier. So I know Zan Adams posted earlier, it's not really a question to say your Spider Spies project was something he was really interested in participating in. But like you, he lives in the north of the country, so outside the range.
Yeah.
What else have we got? Victoria Burton asks, are you using photos of the spiders on, for example, iNaturalist and their interactions there? It's straightforward, although labor intensive, to tag the plant species they're on and what they're eating and extract the data.
So if I have enough time, I will be. I'm going to look into working with master students in this next academic year to get through it because it is a lot of data. Even if I just have it down to the UK, it's thousands of images on iNaturalist. But I can't remember. There is another photo recording scheme which is eluding me, but I have data from another one that's in the UK. It might be the British. I cannot remember the name of it, but it is something that I am going to incorporate. It will just be very labor intensive.
Yeah, I think we'd encourage you if, and I'm not in any way suggesting it's not as reliable, but the spider recording scheme, which we administer, historically it wasn't one that used photos, but as we've migrated more towards iRecord, which obviously is run by CEH,
Mm.
there's of spider records at least more than 1.4 million records now on iRecord in the spider recording scheme, and of course they extensively use, as do we, for verification photographs. So perhaps that's what you were thinking of, Rosie. So
Yeah.
iRecord or our own, which is embedded in there. Fantastic. Well, we haven't got any other questions. So Rosie, once again, many thanks for stepping